So welcome to the 9th lecture of Biology 107, everyone. So in this lecture, we're going to finish up cellular respiration by talking about the citric acid cycle and oxidative phosphorylation. So approximately chapters 6.9 to 6.16 in your book. So to start off this lecture, I'm going to have a question here. So this is question number 1. This is sort of review from the last lecture we had on glycolysis. So how many net ATP are made during glycolysis? A1, B2, C3, D4. So please answer your question on Blackboard. So to answer this question correctly, you need to know the definition of net. So net is essentially how many ATP are made minus how many are used. In this case, 4 ATP are made per glucose molecule, and 2 are used during the energy investment phase. So it's 4 minus 2, or 2 ATP. So the correct answer here is B. So the major point of this question is just to teach you the definition of net. So it's how many ATP are made minus how many are used. And really the only process in cellular respiration where we use this word net ATP made is glycolysis, since that's the only process that actually uses ATP. So just be aware of that net ATP means how many are made minus how many are used. So here's a figure that's probably pretty familiar to you since we looked at it in the last lecture. So from last time, remember we studied glycolysis. So this is all about going from glucose to pyruvate. So for this lecture, we're actually going to be covering the rest of this. So we're going to be looking at pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. So essentially, we're finishing up the rest of cellular respiration in this lecture here. Here's also a little bit of review. So this is the overall summary of glycolysis. So here, remember, we start with glucose, and we end with 2 pyruvates at the end of this process. So we start with a 6-carbon molecule, which is glucose. We split it in half, and we get 2 molecules of pyruvate. So essentially 2 3-carbon molecules. We also have the energy investment phase, where we invest 2 ATP. And we have the energy payoff phase, where we generated 4 ATP total, as well as 2 NADH electron carriers. So it's just a quick review of glycolysis. If you're a little bit fuzzy on this, I recommend going back to the last lecture and rewatching that. So here's another little bit of a review question. So this is question 2 for Blackboard. So the energy stored in glucose for cellular respiration is in the protons, neutrons, electrons, or ions. So remember that glucose stores its energy in the electrons. So those electron carriers, that NADH that we saw, that now has a lot of energy now that it's picked up some electrons from glucose. So the correct answer here is C. Glucose stores its energy in electrons. So don't forget that. That's going to be a very important information as we're going to see in oxidative phosphorylation later in this lecture. So again, today we're going to be focusing on pyruvate oxidation, the citric acid cycle, as well as oxidative phosphorylation. So those are the 3 processes we're going to study. And just a reminder that these 3 processes happen in the mitochondria. So both the mitochondria of this animal cell right here and this plant cell right here. So that's where we see pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation. Also a reminder, in both glycolysis, and as we're going to see in the citric acid cycle, those produce some ATP via substrate level phosphorylation. They also generate electron carriers, which are a little bit covered up by that circle. But as we're going to see, the majority of ATP generated in the cell happens during oxidative phosphorylation. So we're going to see a ton of ATP generated in that step once we get to that in this lecture. And that, again, is going to have to do with these electron carriers NADH and FADH2. So here we have pyruvate oxidation. So as the name suggests, we're going to see pyruvate oxidize during this step. So remember pyruvate, which is on the left-hand side of this picture, is that 3-carbon molecule we ended up with at the end of glycolysis. So essentially, we can see how glycolysis is now linked to this step. And we're going to see the ending at molecule, acetyl-coenzyme A, is actually going to be involved in the citric acid cycle, which is the next step of cellular respiration. So the point of saying all that is just to remind you that all these steps are linked together. So pyruvate produced during glycolysis is going to play a role in pyruvate oxidation, and then acetyl coenzyme A, which is the end molecule of pyruvate oxidation, is then now going to play a role in the citric acid cycle. Okay, so pyruvate is a 3-carbon molecule. You can see that because it has 3 black circles. And we end up with acetyl coenzyme A, which has 2 black circles and also has this coenzyme A group added to it. So the first step in pyruvate oxidation is removal of carbon dioxide. So remember, we breathe in oxygen and we breathe out carbon dioxide. So the carbon dioxide that we breathe out is actually being produced in this step. So this carbon dioxide right here is actually what we exhale. When this carbon dioxide is removed, it's taking a carbon from pyruvate. So again, pyruvate is a 3-carbon molecule. It then produces acetyl coenzyme A, which is a 2-carbon molecule. So essentially, by removing carbon dioxide, we've removed 1 carbon from the 3-carbon pyruvate, and we end up with 2 carbons at the very end of pyruvate oxidation. In the second step, it's important to remember, again, this step is called pyruvate oxidation. So pyruvate loses electrons. So remember, Leo goes Gur, lose electrons oxidized, gain electrons reduced. So if pyruvate is oxidized, something has to gain electrons or be reduced. In this case, our electron carrier, NAD+, picks up those electrons and becomes reduced. So this is step 2 of pyruvate oxidation. And again, Leo goes Gur, gain electrons reduced. You can see NAD+ has gained 2 electrons and now is NADH. So it has become reduced in this step. So essentially, pyruvate oxidized, NAD+ reduced. And the final step of pyruvate oxidation is you have this coenzyme A come along and it attaches itself to this acetyl molecule to become acetyl coenzyme A. So again, essentially go over this one more time. Pyruvate loses carbon dioxide, so it removes 1 carbon from the 3-carbon molecule to become a 2-carbon molecule. It then becomes oxidized, so it loses electrons and reduces NAD+, which becomes NADH. So that NAD+ gains electrons, pyruvate loses electrons. And lastly, we have coenzyme A attached to that 2-carbon molecule to make acetyl coenzyme A. So as we're going to see, this acetyl coenzyme A molecule is going to play a huge role in the citric acid cycle, which is the 3rd step of cellular respiration. But before we go there, here's question 3 for Blackboard. So what is reduced during the pyruvate oxidation step? A, pyruvate, B, coenzyme A, C, NAD+, or D, acetyl coenzyme A? So in this question, you're looking for something that's reduced. So it's important to remember, Leo the lion goes Gur. So lose electrons oxidized, gain electrons reduced. So here we need to find something that's gaining electrons. Also a helpful hint is remember this is called pyruvate oxidation. So from this name or title, we know for sure that pyruvate is being oxidized. So pyruvate is definitely not the answer here. And if you just look closely, remember we have our electron carrier here, NAD+, and it's gaining electrons. So here you can see NADH now has 2 electrons attached to it. So here it's been reduced, it's gained electrons. So really the point of this question was to help you remember Leo goes Gur. So remember, lose electrons oxidized, gain electrons reduced. So next we're going to talk about the citric acid cycle. This is sometimes also known as the TCA cycle or Krebs cycle after the person who discovered it. Again, we're going to produce electron carriers, as we're going to see, as well as some ATP by substrate level phosphorylation. Also, just like pyruvate oxidation, this step takes place in the mitochondria. So here we have a diagram showing the reactions that take place during the citric acid cycle. So just like glycolysis, all these reactions are catalyzed by enzymes. So there are enzymes that are making these reactions more favorable so that they take place. In pyruvate oxidation, we talked about pyruvate being converted to acetyl coenzyme A, So you can see acetyl-CoA is actually entering into this reaction. The first thing that actually happens, though, is the coenzyme A gets removed. So now we just have a true carbon molecule just by itself entering into the citric acid cycle. And if you're curious why the coenzyme A gets removed, we can talk a little bit about that in class. You can come ask me after lab and we'll discuss why this actually takes place. And it's a little bit of an odd step. So why add coenzyme A if the first thing you're going to do is remove it? So definitely see me in lab if you're confused about why that happens. But for the purposes of general biology, we won't talk about it too much. So here we have a 2-carbon molecule, just acetyl by itself. Next, that's going to meet a 4-carbon molecule called oxaloacetate. So 2 carbons plus 4 carbons makes a 6-carbon molecule. That 6-carbon molecule we call citrate. So you can think of the citric acid cycle getting its name from this molecule citrate. So that's why it gets the name the citric acid cycle. So really the first molecule that's formed is citrate here, so that's 6-carbon molecule. In step 2, we have a reduction of NAD+ to NADH, and we also have carbon dioxide leaving. So just like in pyruvate oxidation where we had carbon dioxide leaving and that pyruvate, which is 3 carbons, being turned into 2 carbons, here we have a 6-carbon molecule with a carbon dioxide leaving, and that molecule is now 5 carbons long. So again, when the carbon dioxide leaves, we're removing 1 carbon from the molecule we're looking at. So here that 5-carbon molecule is alpha-ketoglutarate. In step 3, we have another carbon dioxide removed. So we're going to see succinate with 4 carbons. Also important to note is that we have another reduction step here. So we have NAD+ being reduced to NADH. We also have a phosphorylation or substrate level phosphorylation event occur here. So ADP is turned into ATP. So in step 3, we essentially have 3 things happen. Carbon dioxide is removed, NAD+ is reduced and gains 2 electrons, and then we have ADP converted to ATP by substrate level phosphorylation. As I mentioned earlier, we see succinate, which now has 4 carbons. So remember in step 3, a carbon dioxide left, so that now leaves us with a molecule that has 4 carbons instead of 5. In step 4, we have another reduction event. Instead of NAD+ being used, here we have FAD. We'll talk a little bit about the difference in regards to how much energy is produced for FAD versus NAD+ when we talk about oxidative phosphorylation. But just know here that FAD is an electron carrier just like NAD+. So after succinate is oxidized and FAD is reduced and gains electrons, we now have fumarate. So we didn't have any carbon dioxide leave in step 4, so we still have a 4-carbon molecule, just now is slightly more oxidized than the succinate we were looking at earlier. That fumarate then has a water added to it, becomes malate, still a 4-carbon molecule. Malate is then reduced, and NAD+ is reduced, malate's actually oxidized, and NAD+ is reduced. So NAD+ gains electrons, becomes NADH, malate loses electrons, and becomes oxaloacetate. So as you can see, this is a cyclical process. So we started with acetyl coenzyme A plus oxaloacetate. That made citrate. That made then alpha-ketoglutarate, succinate, fumarate, malate, and we ended up back at oxaloacetate. So that's why it's called a cycle. So it keeps on going round and around. So the products we start with, we also end with as well. So that was quite a bit of content right there. So this is also a figure from your textbook. This is a summary of the citric acid cycle. So this is more along the lines of what I want you to know. So again, major inputs, major outputs. I don't want you memorizing each metabolic intermediate in that pathway. So I want you to know the major things that happen in the citric acid cycle, not the little tiny details. So again, the overall picture is we have a 2-carbon molecule coming in. That means with a 4-carbon molecule that makes citrate, which is 6 carbons. And then through a bunch of enzymatic processes, we have 2 carbon dioxides leave, 3 NAD pluses reduced, 1 ATP made, and 1 FAD reduced. At the very end of the process, we end up with oxaloacetate, our 4-carbon molecule, which again is going to react with a 2-carbon acetyl group. So that is the major process of the citric acid cycle. So here is question 4 for Blackboard. So how many times does a citric acid cycle go around per molecule of glucose? So a very important note here is that I'm asking per molecule of glucose how many times a citric acid cycle goes around. So per molecule of glucose. So remember in glycolysis, glucose produces 2 pyruvate molecules, not just 1 pyruvate, 2 pyruvates. Each of those pyruvates then goes through pyruvate oxidation, and each one of them can enter into the citric acid cycle. So essentially, per molecule of glucose, we get 2 pyruvates, 2 acetyl coenzyme A's, and the citric acid cycle then goes around twice. So the correct answer here is B, so 2 times. So it's very important for exams and other types of assessment that you're aware of the difference between per molecule of glucose and per molecule of pyruvate. I'm asking 2 very different questions depending on which one of those I prompt you with. So per molecule of glucose, the citric acid cycle goes around twice. Per molecule of pyruvate, it only goes around once, because that 1 pyruvate can only produce 1 acetyl coenzyme A, can only churn the citric acid cycle once. So please be aware of that when I'm asking questions. Per molecule of glucose is very different from per molecule of pyruvate. So next we're going to watch a YouTube video that's going to show you exactly what oxidative phosphorylation is. So again, oxidative phosphorylation is the last step in cellular respiration and also where most of the ATP is produced. So this video is going to be a little bit advanced, but it will give you a good idea and a good foundation of what exactly oxidative phosphorylation is.
The electron transport chain is a series of protein complexes embedded in the mitochondrial membrane. Electrons captured from donor molecules are transferred through these complexes. Coupled with this transfer is the pumping of hydrogen ions. This pumping generates the gradient used by the ATP synthase complex to synthesize ATP. The following complexes are found in the electron transport chain: NADH dehydrogenase, cytochrome BC1, cytochrome oxidase, and the complex that makes ATP, ATP synthase. In addition to these complexes, 2 mobile carriers are also involved: ubiquinone and cytochrome C. Other key components in this process are NADH and the electrons from it, hydrogen ions, molecular oxygen, water, and ADP and PI, which combine to form ATP. At the start of the electron transport chain, 2 electrons are passed from NADH into the NADH dehydrogenase complex. Coupled with this transfer is the pumping of 1 hydrogen ion for each electron. Next, the 2 electrons are transferred to ubiquinone. Ubiquinone is called a mobile transfer molecule because it moves the electrons to the cytochrome BC1 complex. Each electron is then passed from the cytochrome BC1 complex to cytochrome C. Cytochrome C accepts each electron 1 at a time. 1 hydrogen ion is pumped through the complex as each electron is transferred. The next major step occurs in the cytochrome oxidase complex. This step requires 4 electrons. These 4 electrons interact with a molecular oxygen molecule and 8 hydrogen ions. The 4 electrons, 4 of the hydrogen ions, and the molecular oxygen are used to form 2 water molecules. The other 4 hydrogen ions are pumped across the membrane. This series of hydrogen pumping steps creates a gradient. The potential energy in this gradient is used by ATP synthase to make ATP from ADP and inorganic phosphate. The ATP synthesis steps you see here are discussed in greater detail in the ATP synthase gradients animation. This animation illustrates 2 full cycles of electron donation. In biological systems, however, many electron transport cycles occur simultaneously, helping to ensure that the proton gradient is always maintained.
Okay, so hopefully you found that video very informative. So now we're going to go through oxidative phosphorylation using a figure from your textbook. So oxidative phosphorylation actually takes place in the mitochondria, just like the citric acid cycle and pyruvate oxidation. So looking on the left-hand side, we can see different parts of the mitochondria actually marked out for us. So it's important to remember the mitochondria itself is a double membraned organelle. So it has an inner membrane and it has an outer membrane. So here we have the outer membrane at the very top of the picture. We have the space in between the 2 membranes called the intermembrane space. So intermembrane, between membranes. So between the inner and outer membrane, we have the inner mitochondrial membrane. So this one right here. And then we have the mitochondrial matrix, essentially a fancy word for the inside of the mitochondria. So again, the mitochondria is a 2-membraned organelle. So we're going to be looking at the intermembrane space as well as the mitochondrial matrix. As talked about in the video, there are multiple proteins embedded in the inner mitochondrial membrane. The video talks about enzymes called NADH dehydrogenase, ubiquinone, and other things like that. Our textbook actually uses simpler names, such as complex 1, complex 2, complex 3, and complex 4. So these make up the electron transport chain. The second part of the oxidative phosphorylation is called chemiosmosis. And this is driven by an enzyme called ATP synthase. So really, oxidative phosphorylation is a 2-part process. First, you have the electron transport chain, and second, you have chemiosmosis. The electron transport chain is all about pumping hydrogen, and chemiosmosis is all about producing the ATP using the pumped hydrogen. So first, we're going to talk about the electron transport chain, and specifically, we're going to focus on what the electron carriers are doing. So electron transport chain first starts at complex 1, as you can imagine. The first thing that happens is that NADH actually drops off its electrons. So remember, NADH has 2 electrons, it's going to drop them off at complex 1. Those electrons, as you know, have a negative charge, and so they're going to attract this positive hydrogen or this positive proton, also as it's known. So it's going to pull that proton across the membrane. So it's going to pull it from the mitochondrial matrix into the intermembrane space. So that's complex 1, also sometimes known as NADH dehydrogenase. Once those electrons are done with complex 1, it moves on to some mobile electron carriers. So electron carriers, just as the name implies, carries electrons from complexes. So here, coenzyme Q, or just Q, is an electron carrier. So it's actually going to pick up electrons from complex 1 and shuttle them to complex 3. Once those electrons reach complex 3, we have more pumping of hydrogen, or protons, across the membrane. Cytochrome C, or CYTC, is another electron carrier. It brings electrons from complex 3 to complex 4. On complex 4, we have more protons pumped across the membrane into the intermembrane space. And finally, those electrons are donated to this oxygen atom right there. So this oxygen is sometimes known as the terminal electron acceptor, since it accepts the electrons at the end of the electron transport chain. That oxygen, once it picks up those electrons, then reacts with some hydrogen to become water. So in addition to creating a proton gradient or a hydrogen ion gradient, we also have water produced at the very end of the electron transport chain, due to the electrons being donated to oxygen and oxygen reacting with protons to make water. So essentially, again, the whole entire point of the electron transport chain is to create this gradient of hydrogen ions or protons in the intermembrane space. As we're going to see in chemiosmosis, this is very important for the function of ATP synthase, which again is the major enzyme that takes part in chemiosmosis. So in chemiosmosis, you can think of diffusion playing a huge role here. So we have a high concentration of protons in the intermembrane space and a low concentration in the mitochondrial matrix. So again, diffusion goes from high to low, so we're going from the intermembrane space into the mitochondrial matrix. Protons, since they're ions, do not pass through the lipid bilayer of the mitochondria. So essentially they're stuck in the intermembrane space. So you need a channel protein to pass through. And that channel protein here is ATP synthase. So in addition to being a channel protein and allowing hydrogen ions or protons to pass through the lipid bilayer, at the same time it's using the kinetic energy or the movement of those protons to generate ATP. So as you can see, for complexes 1, 3, and 4, you have protons being pumped into the intermembrane space, but for ATP synthase, you have protons coming back into the mitochondrial matrix. As those protons flow back down their concentration gradient into the mitochondrial matrix, ATP synthase actually churns. So it literally does churn, and that sort of cranking or kinetic motion of ATP synthase actually generates the energy to convert AGP into ATP. So we have the ADP plus an inorganic phosphate becoming ATP. And again, it's this transfer or transport of protons from the intermembrane space into the mitochondrial matrix, which cranks or churns ATP synthase that generates the kinetic force necessary to turn ADP plus inorganic phosphate into ATP. So sometimes I like to think of ATP synthase almost being like a dam. So in a dam, you have a lot of water at the top, and then you have some sort of structure that prevents the water from flowing. Here, that structure is like the lipid bilayer right here. So a lipid bilayer is preventing those hydrogen ions or protons from flowing down their concentration gradient into the mitochondrial matrix. So in the dam, you actually have holes. So a hole right here, like this one, allows for some water to pass through, and as that water passes through, it churns the turbines and generates the electricity. This is the exact same thing with ATP synthase. We're forcing the hydrogen ions or protons to flow through ATP synthase and churn the turbine, or churn ATP synthase and generate energy. Here, the energy, instead of being electricity, is the energy needed to convert ADP plus our inorganic phosphate into ATP. So again, I like to think of ATP synthase almost being like a hole in a dam. So it's allowing protons to pump through, and as that happens, it churns the turbine or churns ATP synthase to make ATP. So ATP synthase is actually a really interesting enzyme. So it's actually one of the largest proteins in the cell, and because of that, we're actually able to use an electron microscope to actually look at it. So we're actually able to generate electron micrographs, just like the one shown below, that show what ATP synthase actually looks like. So here we have a top-down view. So you can see ATP synthase has a sort of circular shape to it. So each of those circles is an individual ATP synthase. So you can see that there's a little hole in the center where actually protons or the hydrogen ions will actually pass through. In these pictures, just have zoomed in versions of that. So you can see ATP synthase has 14 different parts, at least to the top half of it. There are also side views, again, electron micrographs. You can see the different parts of it. So it has this head and tail, as well as this part linking it together. Here's a sort of grainy picture, but same exact thing. We have a channel, and we have sort of a sidearm right here. I think they also have electron micrographs also showing it actually churning, so it shows the churning action as protons pass through that channel right there. So again, ATP synthase is a very cool enzyme because it's so large. It's actually one of the few that we can actually visualize with electron micrographs. So before we move on to the next topic, I want to stop and revisit the electron transport chain just for a few more moments. Specifically, I want to talk about this FADH2, which is another electron transport molecule, just like NADH. As opposed to NADH, which drops off its electrons at complex 1, FADH2 actually drops off its electrons at complex 2. As you can see, complex 2 is not a channel protein, so it doesn't actually pump hydrogen ions or protons across the membrane. Once it donates its electrons to complex 2, those electrons get shuttled to a ubiquinone or Q and goes through the same process as NADH electrons. So the major difference between NADH and FADH2 is the complex they use, and also the fact that FADH2 does not create as strong a proton gradient as NADH. So FADH2 actually generates less ATP than NADH does comparatively. So next we're going to talk about other pathways in cellular respiration and metabolism. So these are other energy sources like fats and proteins, which can also be used in cellular respiration. And we're also going to talk about fermentation and lactic acid production. So these are specifically pathways that are used when oxygen is low. So now we're going to talk about different types of energy sources. So as you all know, it's very possible to live off a high fat or high protein diet. So fats and proteins can provide all the energy or ATP we need to sustain life. So unlike carbohydrates, which enter into glycolysis at the very beginning, as you all know, fats and proteins enter into different parts of cellular respiration. So the glycerol part of fats, in particular, can be converted into G3P, which is a metabolic intermediate of glycolysis. The fatty acid portion, though, can be converted into acetyl-CoA, where it can then go on to the citric acid cycle to produce ATP. Proteins, though, are a little bit more complex, so depending on the type of amino acid, it can either become pyruvate, acetyl-CoA, or be converted into an intermediate of the citric acid cycle. The actual amino group of the amino acid, though, is actually excreted via urine. So as you all know, there's a high amount of urea or nitrogen actually in our urine, and that's because all the broken down proteins, which are used to generate energy, are actually being urinated outside of your body. So the flip side of this is you can actually use metabolic intermediates to generate fats, proteins, and carbohydrates. So for something like acetyl coenzyme A, you can actually combine that with an amino group to form certain amino acids. Pyruvate as well can be used to generate certain amino acids. Acetyl coenzyme A can generate fatty acids, and G3P can generate glycerols to make triglycerides. So as you can see, we can use different metabolic intermediates to generate different biomolecules of life. It's also important to note that ATP is needed to actually do this, so it requires an input of energy to actually generate these biomolecules. So whenever possible, of course, the cell would like to get those molecules from the outside environment, but if it needs to, it can generate some of them via ATP and these metabolic pathways. So now we're going to talk about anaerobic metabolism. So this is the conditions where you have low oxygen. So maybe if you're working out or something like that and you're breathing really hard, you're not getting enough oxygen, this is what your body does. So as usual, we have glycolysis happening. This produces 2-pyruvate, 2-NADH, and 2-ATP. So really the issue here is this NADH. As you know, in oxidative phosphorylation, those NADH will be given a complex 1, and they will lose their electrons, and NADH will be oxidized. So essentially, because you're not having enough oxygen in the system, you have an increase in NADH present, and you have a decrease in NAD+. So when you run out of NAD+, glycolysis cannot happen anymore, so you're not able to produce these 2-ATP anymore. So because of that, the high amounts of NADH is really a problem here. So we need some way of recycling them, and that's through the production of lactate. So as you can see here, we have an additional biochemical pathway where we produce 2 lactate molecules. In the process, NADH gets oxidized to NAD+, and can then be recycled back into this glycolysis reaction right here. So through these means, the human body is actually able to produce ATP even in conditions where there's low oxygen. So this is anaerobic metabolism, specifically in mammals. Anaerobic metabolism can also happen in yeast, but it's slightly different. So again, we have glycolysis happening as normal, produce 2-pyruvate, 2-NADH, and 2-ATP. The major difference here is instead of lactate being produced, we have 2-ethanol being produced. So as you all know, yeast can be used for brewing, so you actually stick yeast in anaerobic conditions and they produce ethanol, which then can be made into beer and other alcoholic beverages. Again, the major point of doing all this is you have NADH oxidized NAD+, and then can be used in glycolysis again to produce these 2-ATP. So yeast can actually live in conditions where there's low oxygen. As you can imagine, though, for mammals and other organisms, it can be very tough to live in anaerobic conditions, which of course is why we need to breathe so we can produce all the ATP we need. However, though, for single-celled organisms like yeast, they can actually live in anaerobic conditions and produce the ethanol we use for beer and other alcoholic beverages. So here's a nice little diagram that we're going to fill out together that I think does a good job at summarizing what we've covered in the last couple of lectures. So at the top, we have cellular respiration, and it has 3 stages. So we're going to fill out the easy one first. So remember, there's only one stage that's a cycle or circle like this. So that's the citric acid cycle, which I'm going to abbreviate CAC. There's also some sort of process that produces some amount of ATP, so not a lot. And that process we know is glycolysis. So both the citric acid cycle and glycolysis produce some ATP, but definitely not as much as this one here, which produces many. So we only know one process that produces a lot of ATP, and that's oxidative phosphorylation. Okay. So next, we're going to go to D. So cellular respiration uses something. So it uses oxygen. And it uses oxygen to pull electrons down the electron transport chain. And this pumps hydrogen ions or protons to create a proton gradient. Next, we're going to look at this part of the figure. So something's oxidized, glucose and organic fuels. So this right here is carbon dioxide. So that's produced. And then we also produce water. So essentially, glucose and organic fuels get oxidized, and this produces carbon dioxide and water. So again, this is a really nice figure that summarizes what we've learned in this lecture and the last one, and so hopefully this makes sense to all of you.